Buongiorno amici! Is it true that we are what we eat? May we even be what our parents and grandparents ate? And what are the implications for our health and the health of our children and grandchildren? In this interview, Professor Randy Jirtle explains how a ghost in our genes may make this possible. I am Dr. Lucia Aronica, and I am on a mission to make the science I teach in the classroom accessible to everybody. If you want to support this mission, please consider subscribing to my YouTube channel and hit the notification bell to get notified when I post a new video. So, let's jump into it.
This on YouTube.
So first of all, welcome. We are now recording for those who can't attend today. As usual, I have many students that can't attend live, so they watch the lecture. And it's thank you very much for being with us, Randy. I am recording this time for YouTube as I'm doing these so I have many guest speakers. So I've decided to record just the lecture part of some of my lectures for YouTube in order for two reasons. First of all, also to promote the course and to create a sort of anthology of the best lectures. For example, today I would like to go with you through the best topics that you have covered during the many times you've been here, but in a way that is more compact, like in 30 minutes, and it reaches also a bigger audience. So the idea, I've done this with a couple of lectures. The first time it was 2 years ago with the Professor Steve Horvat when he developed the epigenetic clock. That was an extremely dense lecture full of slides. And, I think I was one of the first to put online such a very intense lecture on with Steve Horvat. He has become in the meantime he has made so many videos, and my channel is a very small channel. I'm not promoting anything. It's just more to be out to put my lectures out there, and I upload the videos when I have the time, so never. But I will make this happen. I have so many lectures recorded from last year, from other courses, and I still didn't upload them. But now I'm thinking, why not? I've gone through all these videos, and I say, I need to make this happen. And with you, I never actually made a plan to record. And I think this is a good time. So I will upload that on
The very first one interview you had of me, we did record it.
Yeah, no, we recorded. We are always recording. But usually it's only for internally for Stanford, and also this time. So to make sure that my students have the big, how you say in English, the big bang for your buck. We will, I think this will also be good for them. So the first part, we will actually maximize our times, because you will cover the best topics you have covered in many years, and then we will not share anymore on YouTube, and we will have 20 minutes where I encourage the students to ask questions. You will not be on YouTube, so no worries about that. So what my recommendation to the students now is to just make sure they turn off their video feeds and the microphone during the recording portion, and then you can go wild and ask all the questions you want to Randy. Yeah, so we will plan to have like 30 minutes, 40 minutes maximum of interview on YouTube. And I mean, on YouTube, I mean, I will record this and then eventually put it on YouTube, but I will prioritize this. I have many videos I will eventually put in on YouTube. And if students want to help me with that, because the problem is that why I'm so slow is that I do it every once in a while when I have some time in the weekend, which is rare, and probably should be doing something else. But
I volunteer to help Dr. Lucia.
Okay. Then we can
take me upon it. Yeah.
Okay, then we can touch that. Then I will be able to. You know, we have so many other videos. But anyway. So anyway, we can start. I will, Randy, I will introduce you just because we are sharing this.
Before we get going, I wanted to share a couple of photos.
Sure.
How do you share with Zoom? How do you do that?
Yeah, there is a button on the bottom. There is a share screen. Okay.
So the screen, the video, the photo that you have on the top, then when you share, that's the one that pops up. Is that?
Yes. If you do share screen and then you open that, yes, you will have it.
Okay. Okay, let's go.
Are you sharing?
No, I'm not sharing anything.
You want
to share? Know how to do it.
I see. No, yeah, that's the right thing. Okay, so please, can you turn off your video feeds just to make sure that you're not in the video, and your microphone is off, and then we will end the interview in 30 minutes and open the discussion to your questions. Okay. So thank you very much. Okay. Buongiorno amici, welcome to another lecture of the Stanford Continuing Studies course, Diet and Gene Expression. I'm your lecturer, Lucia Aronica, and today our very special guest is Professor Randy Jirtle. Randy is Professor of Epigenetics in the Department of Biological Sciences at North Carolina State University and Senior Scientist in the McArdle Laboratory for Cancer Research at the University of Wisconsin. Randy has made breakthrough discoveries in epigenetics, an exciting field of science that explains why our genes are not our destiny and how the environment and our lifestyle choices can affect our health and that of our children and even grandchildren. For these discoveries, he was nominated Times Magazine's Person of the Year in 27 and featured on the Nova television program on epigenetics entitled Ghost in Your Genes. Randy, thanks for being with us today.
Thank you very much for inviting me.
Oh, yes, it's a pleasure. You have been a guest speaker in this course for many years, and you have delighted many of my students, but every time feels like the first time. So before going in the details of your discoveries and the paradigm shift that they have brought about in the way we think of our own identity and health, I would like to ask you, who's the ghost in our genes, and how did you decide to become a ghostbuster?
Now that's an interesting question. I think a lot of the people that started in the field of epigenetics, I got into the field of epigenetics back in the 1990s, which, you know, it seems like a long time ago now. But people that got into it at that time, many of them got into it kind of by accident. And they weren't trained like people are now in the field of epigenetics. I mean, many universities now have, well, you have lectures in it, and there's many people are trained in that field directly. But many of this older people like myself, the vintage people, didn't get trained in this field. We fell into it by accident. And the way I got into it is that we were studying a gene called the insulin-like growth factor 2 receptor. And we identified that back in the early to mid 90s as being a liver tumor and tumor suppressor gene. And actually, it's now been shown to be function as a tumor suppressor gene in a variety of tumors, probably almost all of them really. And that was exciting, but what happened right at the very same time, and then I want to share now, and so I hope I don't mess this up because I haven't done this before, a painting that you just sent to me, and this is how I actually got into the field of epigenetics, through this person. I think a lot of people in science, if they look back on it, they get to basically Y's in the road, forks in the road. And I had three major ones, and this was the last one that I encountered, and up until now anyway. And that was because of this person here who is a scientist. Host disabled participant screening sharing. What's that mean?
I will enable you the ability to share your screen. Probably you need to, sorry about that.
Yeah, normally I don't share things because of the potential problems like this, but I think it's
Oh
worthwhile.
yeah, so it's worth sharing. You should be able to share. I've sent you this painting yesterday, and
Yeah.
it's a painting of a common colleague.
Can you see that now?
Yes, we can see that. Yeah.
This is a painting of a person that was involved, well, not involved, but absolutely determined my getting into the field of epigenetics. This is Denise Barlow, and unfortunately, a few years ago, she died. And this is a painting of her. Incredible scientist. And she, at that time, back in the early 90s, was the first one to identify this gene, the IGF-2 receptor, as being genomically imprinted. And this is the very first gene that was identified in any mammalian system to be imprinted. And lo and behold, it happened to be the very same one that we showed was a tumor suppressor gene in a variety of human cancers. Now, why this was so incredible to me is the fact that genomic imprinting is an epigenetic phenomena that silences one copy of, of a gene. And it's silenced, these genes are silenced in a parent of origin manner. So for the IGF-2 receptor, the silenced copy is the one that's inherited from the father. The mother's copy is the one that functions. But nevertheless, the important thing is that you've taken basically a gene that on the X chromosome, I mean, not X chromosome, on autosomal chromosomes. And this, I think in humans, is on 6q, that's its location down near the telomere, and made a gene that has a backup. In other words, usually two copies work, and made it into a gene in which only a single copy works. So there's no backup for this gene. And if you think about it, this is really incredible since it's functioning as a tumor suppressor. So the question that I had, and I think a lot of people had, including Denise, because she actually emailed me right after we published our paper about it being a tumor suppressor, because by accident, then we had identified the very first imprinted tumor suppressor gene. And I thought this was so incredible that Mother Nature on purpose would turn off a copy of a perfectly functional gene that functioned in basically stopping cancer formation from occurring. It was bizarre and almost not understandable. The only way you can really understand it to some degree is to look at this evolutionarily. You can't look at it from the standpoint of cancer formation in individuals. But evolutionarily, it makes sense. At the individual level, biologically, from your standpoint, it makes no sense. But that's what happens, and that's what's the case. And there's a number of these now that have been identified. And when I saw that this was an epigenetic phenomena that was turning off a perfectly good tumor suppressor gene, I came to the laboratory and said, we're taking our whole laboratory into the field of epigenetics because it's fundamentally so important that we just can't miss out on getting into this field. The problem is that at that point, I really didn't understand how I was going to do it, but I knew that was what I wanted to do. So this is the person that totally changed the direction of our laboratory. Now, once we knew that this tumor suppressor gene was also imprinted, we got into the field of genomic imprinting early on. And there are regulatory elements that control the expression. So the very first thing we wanted to do and determine is whether the environmental factors could alter these regulatory elements and in a way that would ultimately give rise to disease formation.
Yeah, no, thank you very much, Randy, actually for sharing these pictures. I've sent you this this painting yesterday because Denise was also a colleague of mine when I was doing my PhD, and I had the pleasure to present my first PhD presentations to Denise. And now we have my university in Vienna has also established a Denise Barlow Award for a new young PhD scientist. So thank you very much.
So you and I are probably the only ones that are listening to this right now that knew her personally.
Yeah, no, she was a wonderful woman, very dedicated to science, and I think it's beautiful that the world is recognizing the many contributions she did to science and the future generations of science. There is a spark. Some of my spark for science comes from Denise. So thank you, Denise, from all of us. You touched some important concepts like genomic imprinting and the role of the environment. Let's break these complex topics down by starting with the Ghost in Your Genes documentary, which tells a tale of two mice, two genetically identical mice, the so-called Agouti sisters. Despite having the same genes, however, these two sisters are very different from each other. One is brown, lean, and healthy, and the other sister is yellow, fat, and with a predisposition to diabetes. So how it is possible that two mice with the same DNA can be so different from each other?
Yeah, now I'm going to share the second slide, which will introduce what I call the Agouti sisters, because even though me personally, if you were to ask me what was the most exciting kind of discovery that we made personally, this was an incredibly exciting discovery, but it probably wasn't emotionally the most exciting thing. Mine was the evolution of genomic imprinting and the determination of the human imprintome, again, back into imprinting. But this was, to me, was just a fundamental question about whether or not the environment could alter regulatory elements and then give rise to disease susceptibility, increase incidence to disease susceptibility or disease formation. So it was a model system to us. And lo and behold, if I would have thought a little bit deeper when we were setting this thing up, we actually discovered, not by accident, but we discovered the mechanism for the fetal origins of adult disease susceptibility was indeed epigenetic. So this is how we did this. So this is the model system that we have, and I'm going to share this now.
This is one of the most iconic examples of how the power of epigenetics to modify the way genes contribute to our physical traits and health traits. So look at that.
Now, can you see that, Lucia? It's coming
We
through.
can, yes.
Yeah, so this is the model system that we use. So as Lucia has already uh, stated, these animals are inbred, so they're genetically identical, but yet phenotypically, they're greatly different. They're both female. They're both 100 days old. So there's nothing different in these animals, except what the mother was exposed to food-wise, nutrition-wise, when they were in the womb. So the animal on the left, the obese animal that has high incidence of, these animals that are yellow have high incidence of getting diabetes and cancer. This is an automatic. If they're yellow, they will have these problems. The mother ate normal mouse chow. On the right-hand side, the mother ate mouse chow that was supplemented in methyl donors. Now, why is that important? Because these carbon with 3 hydrogens that are brought down and physically attached to cytosines when they're in a CG type dinucleotide position. And they're involved in regulating the expression of genes. So this is one biochemical mechanism by which genes are regulated in their expression. And this is how you can get a single cell ultimately developing into 260 different cell types, because in each cell type, they're running different programs. So in effect, I think of the genome as being comparable to the hardware of your computer, the physical machine, and the epigenome as being comparable to the software that tells the computer when, where, and how to work. So you can think of, and I come out of nuclear engineering, computer science background, so I think of everything in physical, very simple ways. You can think of a cell as being a programmable computer. And that means then that if you took 260 Mac computers, and you can tell which type of computer I like using, and every one of those is running a different program, that would be comparable to every cell in your body running a different program. They're all doing something different, but they're all running on the same hardware, which means that all the DNA in all of our cells is the same, but the programs that are running in all those cells is very, very different. That's the epigenome. So in this study, when Rob Waterland came into the lab out of Cornell, he was a nutritionist and still is a nutritionist. And he wanted to look at the role of nutrition in altering the expression of imprinted genes. That's originally what we were working on. And we talked a lot about it, and knowing, both of us knowing, being that we're scientists, that scientists are skeptics, we said to each other, we've got to get a model system to address this question, because even if we find a 10 to 20% difference in methylation of an imprint regulatory element, people that have primarily genetic backgrounds are basically going to throw up that this is of no biological relevance. You can just hear it. You don't even have to state it. You know this is what's going to happen. So I said, we've got to have a system where the epigenetic changes are connected to a very visible phenotype, and then ask the simple question, do environmental factors alter that epigenome and therefore give rise to variations in the phenotypes in the offspring? We knew that all these marks are laid down early in development. So that's why we took a system where we were looking at marks that were placed right after fertilization, when all the business is taken care of, and as a consequence during development, and as a consequence, the easiest place in which you can mess it up with environmental factors. In this case, we were hoping actually with increased methylation, that model system, that we would end up shifting the offspring distribution towards the brown and to the healthy animals. Because in this model, there's a transposable element upstream of the agouti gene that when methylated, you go back to basically the normal expression, developmental expression of the agouti gene, and you get brown animals. If that region is unmethylated, it runs off an alternative promoter in the transposable element, and you get yellow animals, which are obese. The reason they're obese is because this agouti protein is inappropriately expressed throughout the animal for all of its life, and it blocks the satiation center in the brain. So these yellow, beautiful yellow animals never know they're full, and they literally eat themselves into obesity, diabetes, and cancer. And when Rob did the study, he clearly showed that packing the system, in other words, the mother's has a lot of methyl groups in her diet, a lot of methyl groups in the hoppers coming in, that he could shift the distribution of offspring coat color to brown, and that was due totally by increasing the methylation and shutting down that alternative promoter in the retrotransposon that was upstream of the agouti gene. That was the beginning, basically, of the whole field of environmental epigenomics, because now, for the first time, there was a memory system that people could understand why something very early in development could give rise to enhanced disease susceptibility very late in our life. And that goes back then, Lucia, to the whole thing of the Barker hypothesis and the epidemiological studies that were done with people that were in utero at the end of World War II. It's incredible. I mean, here our work in 1990s or 2003 is bringing us all the way back to 1944 at the end of World War II.
This is incredible, Randy. I really believe that the power of this experiment is really to create like a stage, a television for the importance of epigenetics to the world, in which you are really truly broadcasting what happens behind the scenes. So with this experiment, basically you were demonstrated that actually nutrition does impact our epigenome by turning genes on and off, and this affects visibly our the traits of the agouti sisters and their health, and specifically, as you mentioned, so the agouti gene is a gene that is responsible also to regulate appetite, and basically because of bad dietary habits, these genes can be turned always on, like in the yellow mouse, and then lead to predisposition to diabetes. Whereas with the good diet, the brown mouse was turning these genes off. Now, talking about this good and bad diet, so you used some a diet supplemented in methyl donors, which are nutrients that provide methyl groups, which are crucial for epigenetic reactions. And these nutrients are folate, vitamins of the B group, choline. So,
And
but
betaine.
yeah, and
betaine.
Yes, betaine is actually very important, and is very rich in beets, right? And but you also used then, so these are where the good things that promote a good shut down, so turning off of the agouti gene. But you also then use some bad exposures, such as EPA, right? So how the two exposures, I think what is interesting, and you show that actually the good diet, a diet rich in methyl donor, can counteract the bad epigenetic effects.
That was the important, most important point of the study with bisphenol A.
Yeah.
Bisphenol A is an endocrine disrupting agent. It was present in plastics, and we were all sort of semi-bathing in bisphenol A back in that era. I don't think it's quite as high levels now, because there's some awareness that these compounds might not be the they might have some problems causing cancer and stuff like this through non-genetic mechanisms. I mean, they're called non-genotoxic agents in the toxicological world. So this is work done by Dana Nolan and I when she was a graduate student in our laboratory. Now she's, has gone on and become very, very really famous in her own right in her laboratory at University of Michigan in Ann Arbor. But what she did then is expose again the mothers to bisphenol A at levels that we're exposed to in our environment, and clearly showed that she now shifted the coat distribution towards yellow, which are the animals that become obese, get diabetes and cancer at high incidence. So in other words, a bad phenotype by the mother being exposed to this environmental toxicant. And then we said, well, it'd be interesting to see because, it's always been said, that food is medicine, right? I mean, that's been said for millennium. And so we said, well, we showed that methyl donors, for example, can shift the distribution to brown. So we asked the question then whether or not in animals that were exposed to mothers that were exposed to bisphenol A, if we then gave them a diet that was like a good diet that was supplemented in methyl donors, the exact same ones that Rob Waterland used initially, could we negate the negative effect, the negative epigenetic effect on disease susceptibility in the offspring? And lo and behold, we brought it right back to normal. So for the very first time again, we showed that indeed, from the standpoint of epigenetics, food is medicine. Very, very exciting, actually. So the question now, is that happen in humans? What are the targets? Are some more resistant? These are all questions that can now be addressed because we do know in animal models that this game can be played and it does work.
Yes, no, this is exciting, Randy. In this course, we have a week dedicated to the environmental toxicant that actually can harm our epigenome. And Professor Michael Skinner is a speaker during that week. And he actually, he has shown that BPA and other environmental toxicants can modify the epigenome in mice across multiple generations, up to the fourth generation. And these modifications are associated also with disease and obesity, and even with glyphosate, for example, which is present in many pesticides and Roundup, for example,
right.
Commercial name. But the point is, yes, as you mentioned, so that epigenetics provide actually a tool to redefine what is a toxicant, because
Right.
BPA is considered as safe in a small, those, it depends on how you measure toxicity. If you look deeper, as soon as you have a tool to measure toxicity, then you can redefine it. Say, actually, this is toxic because it's damaging your epigenome, and these modifications.
But also, Lucia, what we have to remember is that even though in these studies we only use single doses, the toxic effect is going to be dose dependent, and we're going to get into that maybe right now. Actually, everything we've talked about thus far, except for nutrition, all these negative, they're negative. They cause bad problems, in the animal models and give rise to diseases. So the question is, are there situations where you can actually get positive effects? I mean, epigenetic doesn't always, from a medical, I think when you're in the medical field, you always tend to think of negative things because you're talking about people that are sick and you're trying to cure and treat things that are bad, and your mind is more on the bad side rather than thinking about the potential good side. And we again fell into this again totally by accident. And is it okay, Lucia, if we talk a little bit about?
Yes, I would like you to talk. I bet you would like to. I would like you to talk about your experiments, surprising experiments, showing that sometimes bad stressors can actually trigger positive epigenetic changes, which can trigger positive also health changes. So actually this phenomenon is known as hormesis, which in the Greek language means rapid motion. So is the rapid change from like a good effect of a bad stressor to actually a bad effect of high doses. So basically the same agent can show like a curve if you look at its effect on our biology from positive effects at low doses, and then suddenly it becomes better at high doses. It happens with things like exercise, for example, is good, is a stressor, which triggers even inflammation. And inflammation associated with exercise is a stimulus for activating cell repair mechanisms that make our cells stronger on sunlight. And even talking about sunlight, so Randy made some experiments testing the effect of ionizing radiation on the agouti mice model. Randy, can you tell us a little bit more about that?
Yeah, I mean, again, I did not go into this study thinking that we would actually determine the mechanism for hormesis, but in reality we did. And we did this because I wanted to close the loop. In other words, everything that we've been looking at thus far was chemical, nutritional, biochemical, chemistry, chemical compounds. So the question that I have coming from a background of radiation biology is whether physical agents not only potentially they alter the genome, we know that for sure because they cause mutations at high doses. But the question was at lower doses where the mutational effects are very, very small and not very high at all. Is there also the potential to alter the epigenome at those low levels of radiation? And we chose ionizing radiation. And the reason we did this is, again, because of my background in nuclear engineering and radiation biology. I think most people would have never, ever looked at physical agents, but it became a real blessing, actually, to us because there's a lot known on the mechanisms by which ionizing radiation causes usually problems. And so we looked at doses, and this is work done by Autumn Bernal when she was in my laboratory. We looked at doses that are used basically in CT scans and chest x-rays at very low levels of x-rays, not the levels that are used for the treatment in radiation oncology departments in trying to cure cancer, not these high doses, but very low levels that you would see in diagnostic radiology departments, not therapeutic radiology departments. Okay, that's where we're working. And again, though, it's thinking about it and talking about it, it's still weird to me that I never really put two and two together properly, because I assumed everybody, when they talked about the dose response, the risk assessment for radiation, they always say there's no safe dose from radiation. So in other words, at 0, 0 is safe because there's no radiation, and any dose that you have, you're going to have a negative effect. That was the dogma at the time. And that's based on purely looking completely, again, only at genetic effects of the environmental factor, which is in this case, ionizing radiation. So Autumn has started this study, and I go into it because it's always amazes me because and she started and did the same thing, and I said, well, it's all going to work out just like we planned. There's no good dose of radiation. It's always harmful. We're going to shift the dose just like with bisphenol A, shift the distribution of the offspring to yellow. They're going to be yellow animals all over the place. And that's bad in this system. Brown animals in this system, that's good. So I said to Autumn, fine, I was getting no information. Nothing was happening. And I said, Autumn, we should be getting some offspring being born now. I mean, do we have any data? And so her quote, I always quote it. She said, we do. She said, and it's freaky. And I said, Autumn, what's freaky? Because that's not a good word when you're in the middle of you're in the middle of of an experiment. I said, what's freaky? She said, we have no yellow animals. They're all heavily modeled or brown.
So basically the ionizing radiation had triggered like a positive
A positive adaptive response, which she then showed, because we know how this system works, through, again, increased methylation at that transposable element, causing the offspring, I mean, basically all of them, to be brown or heavily modeled.
at low
And
doses,
low doses. So then we had to do a dose response curve, and we did up to the point where you start seeing probably mutations enough that you're killing, because you do get mutations. There's effects on the genome, but there's also effects on the epigenome. And at low levels, the effects on the epigenome as far as animal health overwhelms any negative effects that you're having on the genome itself. It's really incredible. So we did see it curve back up. And it looks like it goes through the abscissa in this model system at about 10 rad or 10 centigrade, somewhere's in that range. And then we know that 80% of the damage that occurs with radiation is through the generation of free radicals. And you can scavenge them up with antioxidants. And when we did this, I said, okay, if this is the case and this is real, Autumn, we should be able to basically eliminate the positive adaptive response that we get from low doses of radiation, which everybody thinks is bad, by using antioxidants, which everybody thinks is good. And lo and behold, that's exactly what happened.
Yeah.
So this is why my guess is that you probably don't want to have a lot of antioxidants when you're exercising, because the free radicals will be scavenged up and you might not get quite the increase in muscle mass and stuff that you would have gotten if you hadn't loaded your system up with gobs of antioxidants. But that's only my guess. This is something that could be studied, though, now, I think.
There are some indications. There are not really clinical trials on that, but there are studies that measured the production of reactive oxygen species in the muscle of people who exercise, and correlated that actually with the positive effects on muscle growth. And that's why there is actually now more and more training coaches are recommending to avoid antioxidants.
Are they really? I didn't even know that, but that's
I
would predict.
They are.
Based on our results, I said, this stuff isn't good for you when you're trying to build. Up until a point, Lucia, where if you got too high, in other words, that we didn't, unfortunately, we're not able to do this. In other words, if you got up to a level that was into the negative effect, could you drop that down now with antioxidants down into the positive adaptive response? I don't know about that.
Yeah, no, I think that we don't know, but I think it's conceivable to think that, many stressors can actually synergize with each other.
Right.
And so there is a stress load, right? The total. Right. And you in your experiment, you have tested just the stress of
and it's clean.
Clean.
Yeah.
But let's say, for example, that there is a person that has multiple stress, bad stress and good stress. The bad stress from work, and then parenting and everything, and then the good stress of exercising. Some people now do fasting, which is also a good stress, but it's a stress. So imagine like this imaginary person who does fasting, exercise, is a mother, works, and then all of a sudden it's just like, wow. So the hormetic curve.
So maybe blueberries are good in that environment.
Yeah, exactly. that at that point, actually, some antioxidant may bring the system back to equilibrium, because even this epigenetic tail, is a tail of homeostasis, which is the language of physiology, right? Is always keeping
Balance.
things in balance. And when you go out of balance, you are in, you need to bring it back to balance. It's called allostasis, where you have your body is trying to do everything to bring things back to balance. So perhaps in that case, antioxidant can be beneficial. So we are
Right.
not saying that antioxidants are not helpful. We're just saying that there is a place for good stress in our life, and probably many lifestyle diseases are actually due to a lack of good stress
Yeah.
from exercise.
And the other thing I want to say about this, though, because it's very specific to the radiation community, but maybe there are people that are watching this. These results demonstrate that radiation hormesis, it does occur. The mechanism for it is definitely epigenetic, not genetic. And it also means then that the linear no threshold model for risk assessment is not correct. I mean, it's that simple. Now, whether or not that is ever going to be looked at, because there's all kinds of other reasons for why people might not want, might want linear no threshold rather than the real dose response that occurs with ionizing radiation. But those are correct facts. And as a consequence now, we have with this crazy agouti mouse system, one using nutrition, the other using ionizing radiation, we have determined the mechanism for the fetal origins of adult disease susceptibility, and we've now determined the overall mechanism for the phenomena of hormesis, which including myself used to laugh about.
No, this is very exciting.
That's the end of it.
Yeah. So, Randy, I would like to ask you one last question, which has always been of interest for many of my students. So, the link between the phenomenon of genomic imprinting and brain health, especially so in terms of explaining potentially the development of some disorders such as autism or schizophrenia. So you have mentioned that this phenomenon of genomic imprinting describes how for certain genes actually we have them turned on only on the maternal copy or the paternal copy. So
Right.
for these genes, we are functionally haploid. We have only one copy that really works of these genes, although things are not black and white. It could be like 40% working from mom and 60% from dad. But anyway, we have this imbalance that is built in throughout evolution for a reason. And when this imbalance is actually perturbed, so we have, for example, more maternal gene expression or paternal gene expression, then this can lead to psychiatric disorders according to this theory, which is called the imprinted brain theory. Can you comment on that?
Well, you pretty much nicely described the phenomena. There's two places where imprinted genes are particularly expressed in a monolithic manner. One is in the placenta, metabolism, basically. And the conflict theory of Hague basically states that the reason these genes evolved was in effect to control the amount of nutrition the offspring extracts from the mother. The father trying to maximize the extraction, the mother trying to dampen it down. And it makes predictions. One of the predictions is this theory makes predictions that paternally expressed genes would be pro-growth and maternally expressed genes would be anti-growth. So if you looked at them from a cancer standpoint, the paternally expressed genes would be oncogenes like IGF-2, which is paternally expressed, is a paternally expressed gene basically in all mammals that have been looked at. On the other side of that is the IGF-2 receptor, which is a tumor suppressor gene, and that degrades. It's not a signaling. It's a degradation pathway for IGF-2. And indeed, that's maternally expressed. So, and that all has held up pretty well. The person who came up, and the other place now where imprinted genes are expressed predominantly is the brain. And so Christopher Babcock came up with what's called the imprinted brain theory, which is basically just an extension of the conflict theory that Haig came up originally based on metabolism. But this has to be is based more on behavior, because it doesn't mean that the conflict between males and females stops after an offspring is born. But for an individual, for example, with let's say a certain set of imprinted genes, to get more nutrition from the mother than someone else that might be sired by a different father, that offspring has to demand more nutrition after they're born. You can't use the placenta in biochemistry anymore to play the game. Once you use the word demand, you're talking about behavior, and it's possible then that imprinting was involved in the brain development and as a consequence, behavioral effects. And what Christopher Babcock predicted is that when you had an overexpression of the paternally expressed genes, it gave rise to the neurological disorder we call autism. And the antithesis of this is when you have an overexpression of the growth inhibitory effects in the brain during development, and that gives rise to schizophrenia. And that every neurological disorder now sits in a bell curve between these two extremes in behavior, autism and schizophrenia. The only way this can be tested potentially now is to have in effect, a better idea of the complete repertoire of genes that are imprinted in humans, and particularly their imprint regulatory elements. And that we have just completed.
Yeah.
So this is now possible.
Yeah, I think this is probably one of the most exciting and promising fields in epigenetics for the upcoming decades. So understanding the
role of genomic imprinting in metabolism, which is obesity, diabetes, that type of stuff, and behavior.
Yeah. No,
it's unbelievable, because now just looking at a few regions, because we're writing the paper now finally, and it's incredible. I mean, areas like Down syndrome, there's a critical region in Down syndrome of 1.5 million bases. Until now, no one knew whether they were imprinted. We now know there's 4 putative imprint regulatory elements within that region. The George syndrome, another one that gives rise schizophrenia, developmental effects, etc., on chromosome, I think 22 rather than 21. 4 more right in the critical region, in genes that have been implicated but were never known to potentially be imprinted or monoallelic expressed, because there's multiple variants of many of these genes. And to go in and look for monoallelic expression is very hard unless you have some physical reason to do it. Well, if you have an imprint regulatory element that's differentially methylated on a maternal or a paternal copy, that's pretty good evidence that maybe you should look at this gene from the standpoint of imprinting, and also look at the potential deregulation of this gene from the standpoint of epigenetic phenomena.
This is absolutely exciting, Randy, and I think it tells a lot about the beauty of epigenetics. I look at epigenetics like a beautiful orchestra that uses the notes of the genes to actually compose a variety of different types of music, depending on environmental stimuli. And even for example, for brain health, we can see how this might help to explain why we see that some brain, so the psychotic disorders also run in family, but sometimes it's difficult to understand their genetic contribution, because it may also be different if you inherit that particular bad gene from mom or dad,
And
depending.
the problem is, Lucia, is it's hard to get the genetics because it skips generations. I mean, it's just a confusing inheritance pattern unless you're really, really looking at it and you have basically offspring, parents, grandparent data that you can start putting together. And now when we have the human imprintome, you can start focusing on literally, Bing, there are 4 of them here, 4 candidate genes now.
Yeah,
Bing.
I'm excited. I think I thought, I was already lucky enough to start actually my PhD when epigenetics was starting to boom, really, like in the early 2000s. And now I think there is even more exciting times to come. So Randy, I would like to.
Can I end with one more thing, one more example, because it's incredible from the area of imprinting, metabolism. DLK1 or PREF1 is involved in the maturation of white fat, which stores energy. So this is the storage depot for energy, white fat, adipose tissue. PREF1 is intimately involved in the maturation of these cells. That is the gene. It's imprinted and paternally expressed. There's another part to this whole thing. Like everything in biology, it's the yin and yanging, right? It's all yin and yang, everything. It's never accelerator without brake. At every darn step, there's brake and accelerator. It's beautiful, actually. Well, the same thing in metabolism. We have brown fat. Brown fat burns energy, burns it. PRDM16 is the gene required for the maturation either into a muscle cell or into a brown fat cell. We predicted in both mice and in humans back in 2005, 2007, using artificial intelligence, that this gene is indeed genomically imprinted. We now know there is an imprint regulatory upstream of the gene, suggesting highly that brown fat differentiation and white fat differentiation are both regulated by imprinted genes in conflict.
It's incredible.
I mean, is this not amazing?
It is. It is. I mean, for me, for a scientist
That's
like me.
metabolism. That's the yin and yang of metabolism.
Well, Randy, thank you very much. I think I will just like to end this interview with one last question. What are your lifestyle actions? I call these lifestyle actions epi actions in this course, just to emphasize that many lifestyle actions have behind the scene an epigenetic mechanism through which they can improve our health. So what are your favorite epi actions?
What do you mean by that now?
Like, what do you do? Like, I know that you are you value the importance of a healthy diet, and also
you're asking me what my hobbies are, or
yes,
what?
Yes, what are your favorite lifestyle actions to improve your epigenetics?
I would say, and you know this, it's riding bicycle.
Yeah.
I mean, I like to maintain a healthy diet and stuff like this, but it's still a blessing to me that I'm able to get out and ride, at a pretty nice clip down the road on my bicycle, and I enjoy that immensely.
I know. You share with me sometimes the loops
right.
that you make on.
They're little loops now because I'm older, so rather than long loops, but they're still loops. So I'm still out there.
I love it. And Randy, I think it's also important what you just said, that for you is a blessing, is a pleasure.
It is a blessing.
And many people think that, pleasure and health are impossible to go hand in hand. I actually believe that the secret really for sustainable health is to meet pleasure with lifestyle actions that can be pleasurable, even exercise, even healthy eating. They are not contradictory, and I think that's the key for long-term happy health. So thank you very much, Randy, for sharing your.
I'm sorry we took too long, Lucia. I mean, we don't have any
No, no,
time to
we will have now time for more questions for the students. So thank
Okay,
you very much.
thank you.
Okay, so yes, we took a little bit more time. Yeah, you can turn on your video feeds and questions, but we will have, if you have time, we can have like 10, 15 minutes of questions if the students have it. Is it fine for you, Randy?
It's fine, yes.
Yeah. And I know Randy is
I'm
always
turning on the
very,
gallery now, so you guys can
yeah.
all turn on your little
And
computers.
Randy is always very generous. Even he shares his email, and if you want to ask him questions, so the question part will not be a problem. And I love this interview because we really went through many topics. So Harshab, you can go ahead and ask your question. You can also turn on the video feed if you want.
Oh,
Ah,
sorry.
the guy with the hat.
Yeah, got some long hair. Thank you for the fascinating talk, Randy, and especially the two rat lab models, really fascinating. And I'm a newbie and trying to connect two topics together, so really love to get your guidance. So using this two rat model, how can I tie it with epigenetic reprogramming? I mean, can any experiments with that work differently with these two rats? And my second question is, I'm sorry, could I ask the second question?
Yeah, let's just go with the first one, right? So you're talking about whether or not you can change in effect the coat color or the disease susceptibility in these animals after they're born, I imagine is what you're talking about.
Correct. So I was really struck when you said that these are two identical rats for all purposes, but are very different as they grow. So if you epigenetically reprogram and age backwards, what do you think will happen to these rats?
Well, it's easier to go forward. In other words, it's easier to make a change early on because you can make it only in basically a very small number of cells, and all the cells then that come from those first one or two cells way back when the decision was made to be methylated or unmethylated are going to be there for the rest of their life unless something comes in and removes those marks. But now the marks got to be removed in a lot of cells because there's a lot more cells in our body as adults than there were when you're talking about one or two cells back in the early days. Dana Dolinoy, my graduate student, is working on this, and there's some evidence that you can use techniques to actually go in, I think involving CRISPR, to go in and actually change these methylation marks such that you could reprogram that area and turn a yellow animal into a brown, or maybe a brown animal into a yellow animal, depending on which way you want to look at it. I think it's doable, but it's not going to be as easy, obviously, as going in the forward direction. The backward direction, like in chemical things, you got a big arrow here and a tiny little arrow this way. And it's a
Okay.
lot harder to make that tiny little arrow give you a phenotype that you want. But that's what people, obviously all of us, are interested in, because this is the way you would treat diseases that are epigenetically involved. The other way, obviously, and now it's also being looked at, is with imprinted genes. If they're involved in the disease phenotype that you're looking at, you think about it, one of the copies is turned off epigenetically. So if there's a mutation in the only functional copy, you will get a disease phenotype. So epigenetic therapy for an imprinted gene is literally just to turn on the epigenetically silenced, and now you've got the problem solved. That is also being done, and there are ways in which this can be done. It's been done now in animal models. In fact, my next door neighbor, Ben Philpott, I mean, you might want to get him on sometime, Lucia, because it's really incredible work that he's doing. I think he works with Prader-Willi syndrome, which is an epigenetic imprinted disease.
Yes.
And he's very much into this whole thing of, in effect, doing what you're talking about. How do you treat imprinted gene diseases epigenetically by turning on the silenced copy?
That would be very fascinating. If you can put us in contact, perhaps just through an email.
I have is, and I'll tell them about it. So that it
Yeah.
would be really quite exciting for your students to, that would address your question a lot more. Now, we're talking, agouti mouse is not imprinted gene, it's a metastable epiallele. And Rob Waterland who did the first study with us back in 2003, has now defined, basically mapped out the metastable epialleles that are present in us. We also have them. The agouti gene is not one of them, but we have metastable epialleles just like mice do. They're
Yeah, and
just
perhaps
different.
you can define that. What is a metastable epiallele?
They have a different fingerprint. And their fingerprint is, if you think about the agouti mouse, yellow and brown, the determination of whether or not that region that controls the coat color occurs very early. So it's either unmethylated or methylated. But once that's determined, every cell in the body has that epi mark. But if you looked at a whole variety of animals, it would basically be 50% of them would be yellow and 50% of them, let's say, would be brown. So their marking varies greatly between individuals, but it's established very early in development. And that is the way you look for these. With genomically imprinted genes, the marks also occur very early. So they have to be present in all cells, all cells in our body, because the marks came in on the gametes. So they have to be present on all cells. And they can't vary between in adulthood between tissues that come from the 3 germ layers, because every cell in the body has to have the same marks, and they have to be differentially methylated. And if you look at all kinds of people, those marks and those regions should be uniform. That's the fingerprint for basically marks that control genomic imprinting. It's a slightly different and subtle, but you can use that information. That information is in our genome. And with heavy sequencing, you can find these regions and map them out. And
Yeah.
Rob mapped out the metastable epialleles for us, for humans. And we have now mapped out basically the imprint regulatory elements, at least version 1.0.
So the metastable epiallele can be viewed as a subset of the epigenetic marks that are written in pen that sometimes I refer to, but probably they might be actually influenced by the environment very early in life.
They will
Right.
be. These are
Yeah.
the I call them epigenetically labile genes that are
Yeah,
basically
definitely.
set very, very early in development. Now, don't get me wrong, there are going to be other epigenetic changes that occur later. But when you're first starting out to try to investigate something and trying to keep things more constant, it's best to work with these that occur early, because
Yes.
you can use any tissue in the body
Yeah.
to investigate them.
Again, as a model, we need a model to
Exactly.
do
We
science,
need
right?
models that are like, in
Yeah.
effect, the agouti mouse.
Exactly. Even, for example, the challenges with our study, Randy, that I still never published actually the result of the study. We are now going to collaborate with Imperial College to pull together the epigenetics data with the metabolomics. But anyway, we see that on a low carb diet and a low fat diet, actually, the epigenetic changes after 1 year are very different, like remarkably different. But the challenge there is that these epigenetic changes can be affected by so many factors, right? In a year, yes, they are significantly different between low carb and low fat diet. So this give us, little confidence if we pick the top most significant changes that probably we have a true signal. But still, challenging compared with studying a model like the metastable epithelial, where you can narrow your focus on things that are
And also these, like with imprinted genes, they're so incredibly important in behavioral development and metabolism that when you find a major change in the regulatory element in these genes, I don't think it's very hard to convince people that you got trouble. So it's not that they're the only things. They're the most extreme things, but they're the low fruit on the tree, and that's what we need to pick right now.
yeah. Anyway, I would like to give you the opportunity to other students to ask questions. 5 minutes left.
Yeah, I have a question. Maybe take more than 5 minutes, but I'm a telomere fan, and it's clearly an epigenetic thing. It clearly affects the diet and it makes permanent changes in the genes and so on in that sense. But I don't see how to put this in the context of everything that you and Randy have been talking about.
Well, I'm not a telomere expert. All I know is that many of the imprinted gene regions, because the imprinted genes, for example, aren't randomly distributed throughout the chromosomes. They tend to be in the telomeres. So there I got the word in. As I was telling you, IGF-2R is right on the tip of chromosome 6q, I think 26 or 27, something like that. And that's true for a lot of imprinted genes. They tend to be out on the tips of the genes, but not all of them, but a lot of them are. But I don't know how this all works because I don't really know much about telomeres at all. Do you have any ideas yourself?
I can just comment. It depends on the interest, Jerry's interest. I mean, I'm also not an expert in telomeres, rather than having done my PhD in Tetrahymena thermophila, which is the model system in which telomeres and telomerase was characterized. So, and I met Elizabeth Blackburn, actually went out for dinner during my PhD. So these are my telomeres tales there. This is my expertise. Elizabeth Blackburn was the scientist who received the Nobel Prize together with the other two scientists for the discovery of telomeres. But anyway, so in terms of now, telomeres have been used also to estimate biological age, because longer telomeres have been associated with a younger biological age. But actually now we know that this association, first of all, seems to be independent from the epigenetic age as measured by DNA methylation clocks. So it's a completely different mechanism. So you don't have correlation with telomere age and the epigenetic age as measured by the biological clock. And also actually the correlation between telomere length and health and even chronological age is closer when people are young, and actually it becomes much more discrepant as they age. There are even conditions, genetic conditions, where you have mutations that increase the telomerase activity, and actually those genetic mutations are associated, not surprisingly, with the increased risk of cancers. So just to say that telomeres don't seem to be like a gold standard in terms of determining your biological age for a number of reasons. Although they are, the telomere shortening can be a hallmark of aging, and it is like telomere shortening, and so things are more complicated that they are just advertised on probably the major headlines, just to say that telomeres are an important mechanism of aging, but it doesn't seem that telomere-based clocks are actually helping us really measure with accuracy biological age or even health outcomes. I don't know whether these
That assumes that the epigenetic clock is the real clock. Maybe the telomere clock is the real clock. So
yeah.
I think the fact they differ is significant, but
Exactly. So the real, what is real is another question because, yeah, exactly. You said it right. They are capturing different signals. And this doesn't mean that, they are useless. They can be useful for different things and in different conditions. For example, you were mentioning what is the real clock. There's no real clocks. Even for the epigenetic clocks, we have now 13 different clocks. Probably we will have other 10, 20 next year. And the question is, what is the best clock? It depends what you want to measure. The best clock so far in terms of measuring chronological age is the Horvath clock 2013. But you may wonder, why do I need a test to determine chronological age with accuracy where we have passports for that? Or, like why we care so much with correlation with chronological age? Whereas perhaps this could be useful for forensic purposes. For example, you find a sample and you want to know the age of the age of the person from that sample. But in terms of, for example, intervention, lifestyle intervention in the clinic, we may want to use epigenetic clocks that are more responsive to lifestyle interventions and therefore can perhaps deviate more from chronological age, right? If they are just capturing that signal of the genome that is more responsive to chronological age, which is probably more a built-in system of epigenetic drift with age, then it's less useful for lifestyle intervention. So again, just to answer your question, all these clocks, epigenetic clocks, are capturing different signals of aging, and they may be useful for different purposes. There are signals, for example, that are more associated with cancer, signals that are more associated with cell senescence, signals that may be more associated with lifestyle changes. So I think the future will be like really to develop like the more precise clock for different outcomes. And telomere-based clock might have a purpose in these things. These are my thoughts. I hope these address your question.
thank you. Yes, very much.
Okay, so that was an exciting discussion. Well, thank you for all those that stayed so long. Thank you, Randy, for your generosity. It was fun as usual.
Yeah, it was great. Thank you.
Okay, thank you very much. I wish you all a nice end of the week, and please feel free to ask questions on the discussion page. See you next time.
Can
Bye.
I get email, please?
The mail. Okay, if I can, I will share. Yes, can I share your email, Randy?
Yeah, that's fine. That'd be great.
Yeah. Okay.
And if you have questions, anybody has some questions, sometimes, like when I was a student, you're a little bashful about asking things, but if you want to email, I may or may not be able to answer them, but I'll try to get back with you at least.
Yeah, Arsha is a very active student who likes to ask a lot of questions. So be prepared to receive a message. Arsha, I love your engagement. So that's why I'm commenting on that. Okay, have a lovely week. Bye.
Bye.